Would like to turn it over to our host, Dr. Danna Schock, who's from Palustris Environmental, and our wonderful presenter and panelists, Dr. Laura Brannelly, University of Melbourne, Dr. John Wood, Pisces molecular, Dr. Alyssa Kaganer from Cornell University, Dr. Emily Hall from Vanderbilt, Dr. Travis Seaborn, University of Idaho, and Dr. Maria Forzan, University of Long Island. And looking forward to a wonderful evening of presentations. And if you have any questions that you would like to pose to the panelists, please put those up in chat. I will be answering those and helping to monitor and make sure that we can get answers to anything that you're interested in.
Before we get started, I know my audio is not working that well, but I just wanted to thank you, Pam, for everything you've been doing to help make this happen. So thank you very much.
Hey, hi everyone. My name's Laura Brannelly. I'm a research fellow at the University of Melbourne. I started working on eDNA during my postdoc at the University of Pittsburgh. So I'll kind of go over some basics of eDNA research and why you might get into it. And then I'll give some examples of some of the troubleshooting that I had to do to kind of get it to work with my samples and the things that I learned along the way. And then I'll talk about eDNA research afterwards. Okay, yeah, so we'll overview eDNA and its uses, the benefits of using it for wildlife pathogens. I'm going to talk mostly about amphibian pathogens and BD primarily, but the techniques can be widely used for a number of different organisms and pathogens. And we'll discuss the methods and the techniques that are important to consider when you're designing eDNA work, and also the limitations of eDNA use in the field. And then I'll use an example of my research of the uses and its limitations for amphibian pathogens. So first of all, what is environmental DNA? Basically what it is, it's extracted DNA from environmental samples to identify a target species. And a common example of this is you look at a water sample and you look for invasive species or some organism's DNA. It picks up the traces of DNA that an organism leaves behind and allows for the detection of the species without actually sampling the individual. And eDNA is used in a variety of capacities. Some examples here are that you can use it to find rare threatened species. So there's been a few examples of using it in the ocean where you can find whales whales and shark species that haven't been identified using traditional methods. And so eDNA has been helpful in that respect. It's used pretty widely for invasive species. In Australia, cane toads are spreading across the country, and there's eDNA programs being implemented in those areas to try and detect first identification of when the cane toad reaches a new habitat or a new area. It has been used pretty widely in pathogen detection for wildlife pathogens, human pathogens, and then agricultural pathogens it's been a really important tool and for that. One of my favorite examples, because it's pretty silly, is that it's been used for looking for the Loch Ness Monster. Loads of water samples were taken from Loch Ness, and unfortunately no Jurassic Age reptile was found, but there was plenty of eel DNA. So maybe we solved that mystery. So some examples of amphibian pathogens that you can use environmental DNA to detect is Batrachocatridium dendrobatitis, so BD, which is a pretty awful amphibian pathogen that mostly infects frogs. We have B. sal, which is the salamander version. It's mostly in Europe at the moment. It hasn't made it to the US yet or other areas around. And so we're monitoring for that. And ranavirus, which is global. we can use eDNA to detect these pathogens in many different variety of habitats. So it's been used in ponds, streams, and even inside bromeliads. In previous studies have shown that the presence of the pathogens can be detected outside of die-off events, which is really important, and even at really low concentrations in the environment. Large quantities of BD-DNA was detected just prior to die-off, so it's possible that you could use it as a prediction method for predicting die-off events. And then ranavirus DNA in environmental samples does tend to correlate with amphibian loads of ranavirus, so it's possibly can be used as a proxy for detecting pathogen load in amphibians. Some of the benefits of eDNA is that eDNA techniques are non-invasive. So this is pretty important, or could be pretty important for things like cryptic species that you can always get your hands on, for threatened species or species in habitats with low density. It can also be used in remote or restrictive habitats and environments where you can't physically get there to sample the animals. So you could potentially use runoff or water downstream to kind of predict where the pathogen might be. And it's possible that it can be used to predict declines. And this is particularly important for biosecurity. So BSAL, as I mentioned before, is not currently worldwide. And we, as disease ecologists and folks interested in amphibians, are testing for BSAL across the US right now, and potentially eDNA can be used to help identify if and when it comes into the country. eDNA can also be used for modeling, so this can be done for transmission or disease dynamics. One possible use of it, if you can get quantity of DNA in the environment accurately, is if you can use it, you could potentially use it as a proxy for force of infection. So eDNA or water filtering could potentially pick up the pathogens that can infect the host. So the amount of pathogen that you pick up might indicate whether force of infection is high or low. One potential issue with this is that eDNA doesn't discern between live and dead pathogen. So you do have to be careful in the interpretation of this, but this is one possibility. And then another use is occupational modeling, which I personally don't have a lot of experience, so we'll leave it to the panel later to discuss this in more detail. Now, eDNA is a fairly new technology, and it's gained a lot of it's been more widespread and people are more excited to use it now that techniques and technologies have advanced. But with this spread of eDNA research, methods across different systems and even within the same field vary quite widely. In BD eDNA work, eDNA extraction methods tend to have a spin column for DNA extraction. There's sometimes a homogenization step, and that's often, it's followed by an inhibitor removal step. So this is kind of like the general way that people with BD eDNA are doing it. But there are some problems with that, and there is a lot of PCR inhibition in many of the studies that have assessed BD eDNA. And so I got into eDNA because we were trying to use it for some sites in Pennsylvania. And I was running into a lot of problems with using it for this method of using a spin column, a homogenization step, and inhibitor removal step. So I conducted an extraction method comparison for my samples. In the literature, comparisons of methods of eDNA are relatively rare. It is or has been assumed that no one extraction method is more robust across different sample types or habitats. And so I tried to do a systematic comparison, and I used 5 different commonly used extraction methods, including ones that were published previously. And what I found with that is that the power soil and power fecal kits, which were mobile, now Kyogen kits, were much better for my pond water samples. And just to give you background in the pond water samples, I was in Pennsylvania, and I don't know if a lot of you have spent a lot of time there, but the ponds are like just basically mud puddles. They're like really dirty, gross water, and there's, you know, the frogs love it, but like it's not great for eDNA. So I wasn't able to get very much water passed through the filter, and I was getting a lot of inhibition. And so this power soil, power fecal, it's basically the same thing. They just have slightly different protocols. Just was able to get really good DNA from those crappy samples. What I found is that it's actually quite a lot higher in detection, 2 to 4 times higher, depending on what the kit I used was. And what I found was that minimum detection level was about 100 zoospores, BD zoospores per filter in this study, which matches up quite nicely with what I am able to detect for swab. For like if you catch a frog and swab that, that's about the minimum detection level for that. So it's consistent using this power soil method. So one thing to note is that there is a step that's an inhibitor removal step that you can add to an extraction method. With this power soil, power fecal kit, you don't need to add that because there are basically no inhibitors that mess up your qPCR. But what I did find when I was trialing it with the other methods is that while it does decrease inhibition, it doesn't eliminate it. And for my trial, there was actually no change in DNA quantity detected when you use the inhibitor removal. So based on that information, it seems like you probably need to get a really good extraction method and probably get rid of your inhibitors at the extraction method level instead of trying to fix it after you've already extracted the DNA. It's also important to note that there are different tacks that you can use for qPCR, which can improve the quantity detected from your extraction method. But I did test this out as well, and still power soil, power fecal kit were superior in terms of the quantity and the detection that I was able to get. Rather than trying to use the environmental master mix on a less sensitive extraction method. So here's some sampling techniques that you do want to consider when you're trying to do eDNA or construct a study that uses eDNA. One of the major things is contamination. When you're picking up DNA from a pathogen from environmental samples, you're looking at such low quantities of DNA that you do really need to be super careful, because you're going to be able to pick up contamination a lot more readily than you would doing other types of DNA extraction and testing. So a thing that you do need to consider is that when you're extracting it, the samples, when you're working with the samples, you do need like a clean room or clean lab space. There's protocols on how to do that, how to make sure that you're going about it properly, but this is something to consider. You want to use bleach, 50% bleach, make it fresh every day. This is way more excessive than what you would ever use with other methods, but this is like really necessary for working with eDNA. And then use disposables that are sterile when possible. Your sample collection methods. So you're going to want to try and eliminate contamination at this step. And this will be things like, right, again, use disposables, don't step in the water before you're collecting the water sample. You want to try and then use negative controls. And so the negative controls will pick up contamination if it's there, or that's what it's supposed to do. You want to try and maximize the volume that can be filtered through your sample. And to do this in a pond, you want to try and get the clearest water possible if you can, because otherwise it's going to clog up your filter and you're not going to be able to pass as much water through it. And then you do want sample replication. So multiple samples per site at one time or across time, over seasons, that sort of thing. You do want replication. For sample processing, you want to try and pass the water through your filter as quickly as possible. You can keep it on ice for about a day if you need to, but it's probably better to do it in the field if you can hook up your vacuum to try and get to do all of that. So just things to consider about sample processing. You do want to try and get as much water per each filter as possible. Aim for over 500, but we don't have a great idea of how much you actually need. And this is probably site, species, pathogen dependent, but you try and get as much as you can, if possible, through your one filter. Sample storage is something that just, there's a bunch of different protocols. And I'm not sure if there's been any real comparisons across protocols. I've used a few of these. You can dry the filter using and store it at room temperature. This is good if you're in the field and you don't have a freezer. You could also freeze the filters. You want to try and get as much water out as possible because water can destroy DNA. So to protect your samples, try and do that. And then extraction and qPCR, this is where you need to consider a number of things. And this, again, has to go into contamination. So we want things like IPCs, which are internal positive controls, to make sure you don't have inhibitors. You want your positive and negative controls so that you can make sure that your extraction method worked for your positive control. And for your negative control, you make sure you don't have contamination in your samples. And then you do want to run your qPCR at triplicate or more. A lot of times it's common in eDNA work to run it in triplicate, and then if one of your triplicate wells turns up positive, you consider that a positive. So there's a number of different techniques, protocols that you can follow. We won't go into, you probably won't get a full protocol from this talk, but these are just some key points to consider. So you do want to know your limitations when you're designing eDNA studies. You want to make sure that your question can be addressed through these eDNA techniques, and if there's potentially a better technique to address your question. One thing that's important to consider is the detection limit for eDNA extraction methods in qPCR. So in a lot of studies that have worked on amphibian pathogens, detection is often correlated with abundance of frogs and also the infection dynamics of the frogs at the time of sampling. So more frogs that are more infected, you're going to be more likely to pick it up in your eDNA samples. If the environment has a lot of inhibitors or dissolved particles or just things in the water that could inhibit your results, you do want to think about this before you get going, because it could limit the utilization of your results. And then with that in mind, your detection limits and what it is that you can think about, what it is that you can actually answer using eDNA, you probably want to do a cost-benefit analysis of using eDNA techniques. So when you're analyzing your samples of water or soil or whatever it is that you're doing, it's generally much more expensive and time intensive per individual sample. But if you're comparing this to sampling animals in the field, you're going to need way more animal samples than you would if you were doing eDNA. So depending on what kits you're using for both of these sample types, eDNA could be much cheaper. It also might be kind of a wash. They might just be equivalent in terms of time and expense. And you can adjust your methods based on your particular conditions for the maximum benefit at the lowest cost. So if you have water that's beautiful and clear, you may not have to do expensive extraction methods to get the same results. And before you go into it, you probably do want to troubleshoot your extraction in qPCR, make sure everything's working. So troubleshooting extraction in qPCR, just make sure that you can actually detect pathogen in your samples and figure out what your limit of detection is before you get going and collect a whole bunch of samples. So an example of how I tested or how I've used eDNA, so I performed a method study where I tried to use eDNA for detection in the field in Pennsylvania and Louisiana. So I had two aims of this project. The first one was to try and understand whether environmental BD DNA correlates with pathogen load in the animal. So I wanted to know whether the load that I detected of DNA from environmental samples was equivalent or it could be used as a proxy to identify load in the animals. So environmental and animals were somewhat similar across sites within a location, across locations, and across seasons. And the second aim of this was to identify whether we could use eDNA techniques as a proxy for force of infection. So I wanted to know whether the amount of DNA of the pathogen that I could collect in the field could be used in models to predict how likely it was that an animal could get infected from being in that pond. I did collect soil and water samples across different environments and also match that up with amphibian skin swabs. I did it in Pennsylvania, in Louisiana, across many sites and multiple sampling points throughout the year. So I'm going to go through Pennsylvania first because this had, so what we found in Pennsylvania is that you actually could use eDNA in filters as a proxy for swab information. So the proportion of samples that were positive in the environment and in the frogs was consistent across the season. And we see some seasonality. I'm going to see if I can get my mouse to work. So we see some sorry, you guys' faces are in the way. So we see some seasonality here. This the gray is the proportion of filters, and the black is the proportion of swabs. So we see some seasonality here, high in the spring, low in June, and then it starts to come back up in the fall. Now with sediment samples, we weren't getting that seasonality, and it wasn't quite as correlated with the swab data. So it looks like we can possibly use eDNA methods to predict the proportion of sites or time periods that are positive for BD. So that's great. It can detect when BD is present in Pennsylvania. Now, if we look at the quantity of DNA detected, here we see that environmental DNA in the swab and in the sediment and in the filter was not consistent with the swab results. So the swab results, the frogs in quantity or infection load was similar to what we saw with proportion. It was seasonal. We have high in the spring, low in June, and then it starts to come back up in the fall, late summer, fall. For the filter samples, we didn't really see that same consistency here. It kind of jumps all over the place, and the same with sediment. So they were not consistent. The environmental samples were not consistent with the animal samples in Pennsylvania at these sites. So we probably can't use eDNA to predict the quantity of DNA in the environment here in this area. Now, when we go to Louisiana in general just has lower infection prevalence than Pennsylvania does. Pennsylvania is there's quite a lot of BD in multiple species. So in Louisiana overall, we saw pretty low prevalence, and the loads were quite low as well in those animals that were detected. We found no positive sediment or water samples across the seasons where we did this detection. And although we did see some seasonal change in swab loads, we see low BD prevalence in the fall, higher in the spring, and then lower in the summer. But this change is actually pretty small. And when we did statistical analysis on these, it turns out that the eDNA presence was equivalent to the swab DNA prevalence. So while we're getting all negative samples, it's still within the error. It's still consistent with the swab samples because it's within the confidence intervals. So from this work and using eDNA to detect BD in the field, found that filter samples and swab samples yield similar results in terms of prevalence of the pathogen overall, but it wasn't correlated with BD load in the animals. Importantly, pathogen presence, there was pathogen presence in swab samples, especially in Louisiana, but this was not consistent for the eDNA detection. And so this is important to note, is that eDNA is possible for presence of, is a possible use of eDNA is for presence of the pathogen, but not absence. And not absence, when there's always an error of missing absence of pathogen, but swab samples are definitely more sensitive at picking up the pathogen than the environmental samples are. In summary, eDNA is a very exciting and new and widespread technology, and there's lots of ways that you can use this technology to answer very interesting ecological questions. But there are limitations, and these limitations need to be considered before you start in on it. There's a detection limit, and you do need to assess what that detection limit is before you get going. It's possibly not really good for predicting pathogen absence in any sort of at any sort of low level. So low level of pathogen, you're not going to be able to pick it up anyway. So you're going to need to use a different sampling method. The volume of water per filter is important. So you do want to try and get as much water through as possible. And if you can't, which is, I mean, there's lots of water from sites that you really just can't pass it through filters, then you need to assess how that's going to impact your limit, your detection limit for those samples. And it's also important that in some studies, like in Pennsylvania, pathogen DNA quantity is not proportional to the infection of the frogs. In some sites, this is true. You can get pathogen DNA quantity to match what's happening with the frogs, but it's not true across the board. And it is really important to troubleshoot before you embark on a project, which I have said a number of times so far, especially because eDNA is new. And if you're new to it, you probably are going to need to give yourself a little bit of time to get up to date with the new practices. Utility of this technique is likely species and site dependent, and pathogen or whatever it is that you're looking for, and the extraction method does matter. eDNA methods should always be used to complement traditional tools, not replace. This is a key point. Yeah, you do want to keep doing those traditional sampling methods. Okay, so that's it from me. We're going to go to panel discussion. I do want to give you some resources. So these are some articles that we all picked as some key points for you to look at. You can email Danna if you want these. And then here's our contact information.
Okay, thank you, Laura. I know my audio is not so great. I'm going to turn off my video and then talk for a bit in the hopes that it'll help a little bit, just so you don't think I'm this disembodied voice out here. So the impetus for this whole presentation was that collectively, all of us on the panel would like to get people thinking proactively about their eDNA related studies. And so this is why we have this diverse group of people here. And so what I've gotten prepared is a list of kind of questions to start things off. And the panel will kind of kick some of these ideas around. And then also I can see lots of people are chatting here as well. And we'll try and fold some of that in too. As you can see, there's a diverse group of people here that use eDNA in very different ways. And so I'm assuming our audience is the same. So I think the panel, we've all seen these questions before, so I'm going to start at the top, just partly, to kind of get things going. And so everybody just jump in when you feel like you're ready to share something, if that sounds okay. And the first question that I wanted the panel to sort of flesh out, and Laura touched on some of these, but I can see from the chat that people have other related questions. So I would like it, if the panel, could we just kick around some practical considerations while collecting in the field? And I know some of our panelists, I don't know, I guess I'll pick on Travis if you feel like you want to do this. I know Travis and I have had some conversations about field collections in the past, and Emily too. So if you were going to make a recommendation for practical considerations, Travis, what would you like us to do?
Yeah, so in terms of starting with practical things to be focused in on, my take-home message there, and I appreciate Laura, she sent the slides around. I was like, please underline this, because one of the things that I think is really important for the field, and it also goes into the lab too, is to really be careful and think about, what your positive and negative controls are before you actually get out into the field, because that's collecting those things and doing those things properly in the field in the lab is something that, unfortunately, if you don't do them right, can kind of doom your study a little bit, in terms of being kind of big red flags on the end of being a reviewer. And so, because we're talking about field studies, one thing I will mention, contamination came up, and so you want to be, for instance, taking your field negatives, after you've collected at a site, and using that to basically check and see if you contaminated all of your samples. You might think, okay, field negative, so I need to run some regular water, but it matters a lot that it's done at the end of the site and not at the beginning of the site, for instance. And so really kind of think carefully about, what's the purpose of each one of these controls and the overall study design. And there's 5 other people, so I'll just kind of pass it off there, because I don't want to go on for too long. So I don't know, Danna, if you want to call out or if I should popcorn to someone else, you let me know.
No, I'll go ahead and start, Travis. So one of the things that I would emphasize, Laura talked about it, how to preserve the samples. Many people have tried to preserve samples or do preserve samples with desiccant beads. That's fine, but don't let the desiccant bead be in contact with the filter. When I get those samples, the filters have desiccant beads stuck to them. And that wreaks all sorts of havoc for me trying to get it into a microcentrifuge tube. So if you're going to use desiccant, which is perfectly fine, at least fold the filter sample in half so that the inside, the environment side of the filter is folded in and is not being rubbed or abraded or stuck to by the desiccant beads. Another thing that we've actually run into is people saying, well, we preserve many samples in alcohol, so we'll just put the filter in alcohol. Well, but that washes everything off the filter. And so now I have to test both the filter and the alcohol. So no, we don't like filters like that. So I think that's one of the other questions that Laura alluded to. When you're getting ready to start an eDNA sample, talk to the molecular biology lab that's going to process the samples and make sure you and they understand how you're going to collect the samples, how you're going to preserve them, and in what form they're going to show up in the molecular biology lab.
Can I follow up? It's Maria. Can I follow up with a comment about ethanol? And I agree with John completely. You need to talk to whomever is going. If you're not going to do the processing yourself, you need to talk to the molecular lab who is going to run it and basically go with whatever they prefer to use. Just a quick comment on ethanol. I worked with someone. I actually provided the chytrid samples for them to try running through a filter and then put half that filter, dry it out and send it as such, and the other half in a little bit of ethanol, not a lot of ethanol, but a little bit of it. And they actually got better results from the ethanol filter than from the dry one. But again, it wasn't swimming in ethanol. It was just a small amount.
Okay.
And as to the problem with transporting ethanol, if it's shipping, I believe if it's under 50 milliliters of total volume, you have no problems, and you would never have more than 50 milliliters if you're shipping filter papers that are just slightly moistened by ethanol. If your problem is driving with it, there's also limits to how much you can have within the vehicle. So it's worth checking that out, but it's not a bad option. I just wanted to add on the ethanol thing. Some people, and I know it's probably no one who's participating in definitely none of the panel, but no one who's participating in the webinar, but just in case, some people use ethanol for disinfection. And that's a bad idea because obviously ethanol will preserve the DNA, so it might inactivate it, but it does not clean as bleach or other substances do. So that was my spiel on ethanol.
I want to talk about just my personal experience with these different storage methods. I personally like the freezer one. You can kind of pre-process before, or like when you get the filter, especially if you do it in a lab, you can get them in tubes and they'll be ready to extract. With the drying filter, like using silica beads, the filters, I guess this probably depends on which filter you're using, but the ones we were using got really brittle and had a lot of like static electricity happening. So it was actually kind of difficult to get them into the tube for extraction. So just for ease of the person doing the extraction, that might depend on what type of sample method you use, sample storage method.
Also, I like silica because I have tipped over my little ethanol vial, in the field, in particular when it's really windy, when you're like trying to like get it, or you have one where the screw cap's not down quite all the way. So, that's my personal bias too, in terms of what works better for me under crummy field conditions.
Yes.
Yeah, and you can just use little coin envelopes. They're really cheap, and just stick the filter in the coin envelope, and then it doesn't touch the beads at all.
Yes, Emily. Yes, we've told people that.
Why don't they listen to you, John?
Yeah.
I guess sort of dovetailing with the field side of things. So we've been talking about actually collecting the samples, but could we back up a step and maybe think about some practical or sort of things to keep in mind when you're actually planning this study? One of the things that I have encountered are people sampling for things at, say, the wrong time of year. There's not a lot of reason to expect water samples to contain frog DNA and BD DNA in February in Alberta. Like, that just doesn't even make any sense. So I guess I'd throw it out to the panel with some sorts of practical things to think about when you're actually designing this study. So we've checked off the box. We're gonna get in touch with our molecular lab so we don't blow up our own experiment that way, but some of the other planning things that people would keep in mind.
I can jump in. Yeah, so I've actually studied seasonal dynamics of disease, so I feel like season is right up my alley. But I mean, just like we're saying, you should get into contact with whoever's running your samples in the lab. You should also contact the people who are on the ground in the field, if that's not you, and figure out exactly, what is your target host and pathogen and when they're most likely to be prevalent or abundant. And, also troubleshooting the actual collection of the eDNA with the folks on the ground. Like Laura said, you want to make sure you emphasize with your field crew to not enter the pond, say to do traditional sampling methods to look for, are the tadpoles there? They want to collect, you want them to collect the eDNA sample before they enter the pond. And then decontamination protocols, of course. I don't think I have to emphasize that to the, I hope the crowd who is aware that before entering new sites, when you're just coming from another pond with amphibians in it, you don't want to be carrying those pathogens between sites. So decontaminating with 10% bleach. So that's store-bought bleach that you've made 10%. That is for gear, but I usually douse everything in as much bleach as possible. And make sure that you rinse that off too of your gear before you're entering the ponds, because that's also not a great thing to add to the ponds, in addition to the pathogens. But yeah, and season, and I'm not sure about time of day, Danna, you put that on the list. It might be worth just talking to your field crew about, time of day that's best for them. And
Sí, ya yo le puse.
But seasonally, I mean, even when you're just sampling for frogs, you don't want to be out there in February in Alberta, let alone their diseases. But maybe that's a good field negative, collecting water samples when you know the host and pathogen aren't there, and then seeing if there's any DNA in your sample. I mean, don't replace your real field negative, which is just tap water run through the filter and ran to see if you can get any DNA. But it's kind of like a field shouldn't be there negative.
I guess this might kind of dovetail. I can see there's lots of conversation in the chat about what is a good pilot study. There's a lot of support for it. So maybe can we just and can everybody share what they think what is a pilot study from the field biology side and the molecular biology side? What is a pilot study? I see all this conversation going on here.
Actually, Karen Goldberg just gave a talk about how to design a good pilot study. I'm hoping that I can find that video and post it on the Canadian Herp website. She was just at an eDNA resource technology exchange. But just to channel my inner Karen Goldberg, definitely what you want to do is from your kind of bigger sample of ponds, try out a smaller subset, collect as much as water as you can from sites that kind of span different environmental factors. So a lot of things will affect the persistence and the degradation of DNA in the environment. So if you have sites that vary in tannins, in pH, in temperature, any kind of pond chemistry data you already have, you already know salinity, just take samples that kind of span that range, and then, test for inhibition. Like we said, you can take that pond water into the lab, like Laura Brannelly did. You add some of the target DNA to the pond water in the lab and actually look at how efficient your extraction and qPCR assays are at detecting those target DNA, because you know how much you added. So you should be able to do the math and figure out how much you find in your qPCR assay.
I think,
sorry, I'm writing notes frantically. That's what's happening for me. So if the panelist wants to jump in, then I'm just making a list of all the stuff to compile. That's what I'm doing right here.
Another thing I wanted to add about pilot studies is they are a good source of information to calculate your sample size. I think Pam asked about, how many samples are you going to need? How many samples should you collect? And I think, as many people have said, there are a lot of variables that will determine that: the site, what you're looking for, what species are present, what prevalence you're expecting. So if you do a pilot study, you might get some idea of how many samples is the minimum that you're going to need to be at least, say, 95% sure that if you don't find something, it's likely because it's not there. I agree with Laura, you cannot swear that if you don't find something, it's because it's not there, because you might just not have detected it. But at least if you have a pilot study that tells you, well, we find it in this very, very, very minute amount, then, or in very few of the samples that we run, then you know that you need to take a lot more samples or look at a lot more sites to be a little bit more certain of your findings. So yeah, pilot studies are key.
For figuring out, for your spiking your water samples to try and figure out your detection limit, I know we were talking about winter samples. That's actually what I used, was like February samples in these ponds. This kind of also might address Chris's question, which is about whether winter sampling might actually be a good time to test for pathogen eDNA, because those frogs are hibernating in the water. At least at my sites, pathogen load on the animals was really low in the winter. So the animals tend to come into spring with really low loads, and then it pops up in load during the spring. So it was a pretty safe bet for me to say that those samples had no BD DNA.
I would just add a kind of cautionary note. I think that's a really interesting idea to think about looking in the winter for different herp hosts. And it's a really great point that you would have kind of lower prevalence of problems with temperature and depending on your location, UV. But I would also encourage you to think a little bit about the source and sink dynamics of eDNA in the environment. And animals that are more metabolically quiet are going to inherently be producing less eDNA. So there's a little bit of a kind of trade-off dynamic there as well. But as a kind of antithetical note, it's also very possible to find things at a time that you might not expect them. And we've found ranavirus eDNA at pretty high titers as late as September, October in some of our vernal pools, which is well outside the time frame of when you would expect an active outbreak to occur. So I just generally, when you're evaluating various ideas for a pilot study, but also your work, I really encourage you to think hard and as holistically as possible about your system, and really focus on your experimental design and not just grabbing water because it's there. It's really tempting, especially if you have something that you're expecting to find seasonally, but being thoughtful up front will more than pay off in the long run.
This is kind of a related point that I'll add to that about when you're planning ahead. One point that's come up with the panel that is one of the challenges is, how do you really know whether or not something is not there or whatnot? And so that's where, if you're planning ahead, even with pilot studies, if you can design something with some repeated measures and take an occupancy modeling approach to allow you to start to calculate out things like your detection probability and start to have a better understanding of your ability to detect something, can be really helpful for informing your bigger, more widespread research project. And I'll say there are a couple of the references that we've sent to Danna that involve occupancy modeling, because it does have some utility, but it does add some additional planning and work up front because of some of the repeated measures requirements.
Up on the slides right now, I just posted some additional resources that you can use for some of these questions we were talking about, especially protocols like the Goldberg Lab resources on these occupancy models. Papers are up here as well.
I have kind of because I see we're at the 1 hour mark, and so I kind of wanted to. This may be a little bit surprise for the panel, but I had a question for you guys. We're thinking about all these pieces and all of these great things. I was wondering if the panel, could you share any applications of eDNA that you have seen recently that caught your eye? Not just Loch Ness Monster, because I mean that's pretty funny and whatever, but are there some applications that have really caught your attention because of their utility or just ingenuity or anything like that? So in your travels, what are some of the recent uses of eDNA that really, really captured your interest? On John, because he looks like he has something that he's ready to share.
One of the things that so far, all of the eDNA work that we've been talking about today is detecting one species. You can actually pretty easily throw in next generation sequencing techniques and go after not only microbiome, but macrobiome detection. So I think this is really the frontier to say, okay, we really want to know in this lake or this particular niche in the ecosystem, who all is there. And if you use NGS techniques, you can say, well, these are all the fish that are there, and these are some of the macroinvertebrates that are there. And yes, indeed, wolves and foxes and whatever are stepping through the water as well, or not. So I would say, think bigger. If we're going to use this for ecological detection, let's get a bigger chunk of the ecology.
Definitely. There's so many people doing metabarcoding for diversity of all kinds of organisms. And I mean, if you only care about amphibians too, you can get the diversity of amphibian hosts along with their pathogens in one sample versus spending hours and hours and hours. I mean, you still need to do sampling throughout the year at different locations of the pond, but if you don't have the field techniques or the, IDing, I mean, amphibians is a little easier, especially in Canada when there's only, how many species? Like 5? No, you guys have more. But, people doing eDNA work with, macroinvertebrates where you really need to spend 6 years studying how to ID all these different mouth parts. Metabarcoding has been super useful.
I would just add to that, again, maybe not necessarily in Canada, but for a lot of our lesser known herp species, there is going to be an issue of acquisition bias in the databases available. So there are a ton of lesser known herp species for whom there are partial, none, or just a single instance of various DNA sequences available in publicly obtainable databases. So having some sort of sense of potentially knowing ahead of time that you are going to need to contribute to a database, do mitochondrial or genome segment sequencing for your targets is a really important way to help flesh out those databases. And there are some really great eDNA metabarcoding databases, I think, particularly for fish that are available.
So I'll bring up a slightly different thing that I'm super kind of excited about, which is just that related to next-gen sequencing and just the continued improvement for some of the molecular techniques. There's kind of 2 topics here that I think are really interesting to think about. And one is actually instead of just focusing on environmental DNA, but thinking about environmental RNA and basically looking at transcription out in the field to get an idea of kind of what's going on there. There was kind of an interesting tree paper that came out about a year ago on that. And then the other topic that I think is really interesting is, as it looks like in some systems, what we're picking up on is actually low quantity, but the lengths of some of the DNA might be sufficient enough to be able to actually do like some pop gen work in the future. So I think that would be super cool, just because that would open up a whole new array of existing analytical and statistical techniques that have been well established that we could apply to these situations where it's difficult to say count individuals, but we could use this to say do effective population size or things like that. And I know Karen, along with her grad student, Megan Parsley, have a chapter about that in the population genomics book that came out recently, I think this year. So that's kind of what I'm excited about, as someone who does like half their stuff doing like landscape genetics, landscape genomics stuff, that it makes me excited to think about.
I'll just jump in here with one thing that sort of I thought was really interesting and exciting, and it's actually listed here on Laura's slide. I thought it was really an interesting application of eDNA approaches to try and improve the situation with the pet trade, because many of the people in the audience here are going to be aware of the incredibly important role that the pet trade has played in translocating pathogens everywhere. And so I thought it was a great example of see a problem, fix a problem. And it's an approach. It's not perfect, but it's way more than was available before those kinds of approaches. So that was one of the applications that really caught my attention just recently, about what a great way to use these techniques to just get at the heart of what's spreading some of these pathogens.
I'm just I was looking through the chat here, and I think I missed a question about false positives and false negatives. So if we, would it be okay to answer that?
Sure. I mean, we should probably wrap up fairly soon for everybody, but you know what? It is a really important concept, and you're right. I think we maybe missed it. Do you want to just pop back to that? I know that's a conversation that the panelists we all had getting prepared for this. It might be a good idea, as long as everybody's got a minute.
Yeah, just I'll try and we can go quickly. So this is like very important to just understand what your false positives and false negatives are. This is true for any DNA work. You're gonna run into these issues if you're working in this space. So for the study that I presented, I didn't get any false positives in the data that was used in the paper, but I was encountering false positives before I changed my contamination protocol. So as we talked about, you need like a clean room and all of that, like clean room to do the extraction. You got to make sure that you're bleaching everything all the time. When I was moving my filters between tubes, I was flame sterilization. I was using flame sterilization before, because that's like what I had always done with swabs. And I was getting false positives. So you do just want to probably be a little bit more aware of that. Just go with bleach, because that gets rid of it. And yeah, false positives, you can detect them with your negative controls, and you want to have a bunch of negative controls at all steps along the way. So in the field, you want negative controls while you're doing the extraction, and then you want different negative controls during your PCR.
I've covered everything that I had on my list. Are there any other questions or anything else that the panel wants to share before we wrap things up?
I wasn't sure, Emily, if you saw the question directed towards you about coronavirus strains or not. Oh, there it is.
So there are different strains of all the pathogens, including BD. As far as I'm aware, there aren't qPCR methods using qPCR primers to look at those different strains yet. Not widely available anyway.
Something important to note about BD, that the different strains do vary in how many ITS copies they have per zoospore. So I would recommend using a synthetic standard in your qPCR protocol unless you actually know the sequence of the local BD strain in your local ponds, which may not just be one, there may be co-occurring BD strains in one pond. So it's best to use a synthetic standard so that you're getting a unit of copies per mil instead of zoospores. And so that's going to change your limit of detection based on the number of copies per zoospore. Well, it's not going to change the limit of detection if your unit is copies, but it will if you're trying to backtrack into zoospore counts.
Yeah, I guess I'll expand on that, Emily. So a lot of the eDNA samples we get positives, and we report copy numbers per sample. And inevitably the question comes back, well, how many animals is that? And I think it's important to realize that when you are doing eDNA testing, we're now talking about molecules, and that's what I'm measuring. I'm not measuring number of animals. And so the question doesn't make sense. And so as you're planning your study and what you're trying to prove or disprove or figure out, make sure that you're good with copy numbers per sample, not number of animals.
Yeah, it's not going to work for a number of animals, even when you have
No.
A mammal or a fish or an amphibian.
No, but you get that question. Oh, you got 10,000 copies. Is that one fish or is it 100 or whatever? No, it doesn't work that way.
I have seen some recent work that copy numbers scale allometrically with body mass, but again, that is, I think, something that would only really be applicable in a very highly controlled and well-studied system, and not necessarily something that's going to be appropriate for a surveillance application.
And it will vary between species, Alyssa.
Right, well, that's exactly my point. Yeah.
For example, fish apparently secrete lots of particles with eDNA, mussels very much less. I mean, they don't have mucus being shed off the outside of a swimming animal, so it's much less.
Even between individuals within the same species, yes, they secrete different eDNA amounts. Not to scare you all away from the eDNA world, but there's a lot of different variables that will affect the secretion, production of DNA.
Very much. So again, as you're designing your study, remember that the data you're going to get is copies per sample, not animals or body mass or anything else yet. And you need to be, that needs to work for whatever your study is aimed at. I suppose I'll throw one last little bit in. A lot of Laura's talk and a lot of worry about eDNA samples is inhibition. By now, we use an inhibitor-resistant TAC environmental master mix, and we have no problem with inhibition at all. I mean, we're not using power soil or power fecal. We're using the standard QIAGEN DNeasy. We run IPCs in all of our reactions. I would say by using an inhibitor-resistant TAC master mix, we get inhibition in 1% or less than or of our samples. So to me, that problem is gone away.
One thing that you mentioned, John, the use of that IPC, that internal positive control, I think that is something that at least some of the things that I've encountered is that step is not included in a lot of studies. And I'm not sure if it's a cost cutting or if people are just making the assumption that, well, they've diluted their water sample and therefore they don't need to use it, whatever. But I heard you just now say that you always use IPC, and I'm seeing a nod from the panel. So you guys are all agreeing. No IPC, you're running the risk of not knowing what's going on. Is that?
You always have to have an IPC in eDNA PCRs. Yep, it's got to be.
I'd like to comment on that, the TAC. Yeah, we have tried that as well, based on your recommendation, John. I tested it with the eDNA kit that I extracted with, and it did increase the quantity of DNA that I was able to detect. But I think inhibitors also kind of mess up your extraction level as well, because all of it's going through a filter, right? So if you're clogging that filter with those inhibitors, you're going to limit the amount of DNA that's going to come through, regardless of whether there's inhibitors or not in your final product. So I think, especially if you have really dirty water, you do want to be careful about that. You want to try and use the best kit available, and then, and that's going to give you a better yield, kind of regardless of whether you use the extra steps of cleaning things up after the extraction level.
Yeah, fair enough, Laura. All I was commenting on is that with the inhibitor-resistant TAC master mixes, then I know there is no inhibition in the qPCR.
Yeah, right, which is important.
Which is important, which is critical. Yeah.
So I will just kind of circle back around then to something that was brought up very early, which is communicate with your molecular biology lab what yes. You're gonna what steps they're gonna take and those things before you
yes.
into a collaboration. Talk about all of these kinds of things, so no surprises at the other end.
Yeah, while you're designing your pilot study, not after you've finished the whole study and send me the samples.
On that note, too, talk to your field crew.
Yes.
Are they entering the pond before they collect the eDNA sample? Are they, actually using a different cup to scoop the eDNA sample, or a new glove, or a clean bottle, et cetera, et cetera? Is it actually feasible for them to get to clean water without stepping in some muddy water first and contaminating the sample? And how feasible is it for them to lug all this equipment up 8,000 feet in elevation or wherever they're going? Communication is key to all teams involved.
Yes. Before, not after the study.
Before you submit the manuscript to the editor.
No, way before that, Donna.
Way before that. There's nothing that we can do then. I think we'll call it there. I see we're way past the hour mark. Thank you for everybody on the panel. I really appreciate all your effort and time to participate in this. Thank you again to Pamela and the CHS for making this possible. That I really am grateful. And obviously, thank you to all the audience and participants. I know many people have had to leave already, but thank you. If anybody has questions or wants more information, you can get a hold of any of us. Hopefully we've given you some food for thought. We want to see everybody just embrace eDNA in the right way, not when you've run into problems at the wrong end.
Yeah.
So
that goes for any papers you try to submit to Danna, eDNA or not. Do a pilot study.
Yeah. All right. Well, I guess, Pamela, is there anything that you needed to tell us or say to us before we all sign off here?
Yeah, I'll just do a final wrap up. So I just want to echo my thanks to Danna for pulling this together and bringing folks from around the world. Much appreciated that you took your time out this evening. The talk will be posted up on our YouTube channel, so you can see the links there too, but I can connect with Danna as well and get information out to you. So do feel free to email me at conference@canadianherpetology.ca. And with that, we will wrap it up this evening. So just huge thanks and reminder, we will reconvene in a couple of weeks, December 6th, for an excellent conference number 2. So I look forward to seeing everybody there, and have a wonderful evening.